Okay, so thank you very much for this opportunity to talk about my research. I'm going to talk about newly discovered lineage of Rhizaria. Rhizaria is one of major eukaryotic lineages or supergroups. And
before you get started, can you put your slides into presenter mode?
I think I did. Is it better now?
No, it's still in the preview or the normal design.
Yeah, let me do it the other way. Sorry about this. Is it better now?
Yes, perfect. Thank you.
So Rhizaria is one of the major eukaryotic lineages, and it includes iconic textbook protists, right, like Radiolaria and Foraminifera, but also many free-living amoeboid and flagellated organisms, as well as some plant and animal parasites. And metagenomic data actually show a considerable amount of environmental diversity in Rhizaria, which is not characterized with cultured representatives and for which phenotypes are completely unknown. So what I'm going to talk about is our attempt, our small step to fill this gap and characterizing a newly discovered lineage, which is amoeboid cells infecting green algae. So let me introduce you to the green algal species, which is called Mucidospherium pulchillum. And these are widely distributed planktonic freshwater colonial green algae, which are represented with small coccoid, pretty like chlorella cells sitting on the branching stalks, which are made of cell wall material. And the whole colony is surrounded with transparent mucus layer, which is often invisible, but has many bacteria attached to it. So Anton Alef and I, Anton Alef is an undergraduate student I had the pleasure to work with. We were looking at plankton of this small freshwater lake and stumbled upon really unusual colonies of Dictyospherium. Instead of having these branching stalls in the center of the colony, we saw this mass of spherical amoeboid cells in the middle. And also the surface of the colony was producing long, thin pseudopodia, which was quite unusual. So when we were looking closely and these infected colonies, we saw that actually the amoeba living in the center of the colony has relatively large nucleus, granulated cytoplasm, and the long thin pseudopodia with granules, which are presumably extrusive organelles. Moreover, as I can show you with this 4 times accelerated video, the whole colony is capable of movement, which is not the case for uninfected colonies. So the whole colony can be gradually dragged across the substratum using pseudopodia. And in the next video, which is unlike the previous one, is not accelerated, it will be played in a normal speed. You can see that actually infected colonies are capable of really fast spontaneous fragmentation, which I'm also not aware of being known for uninfected Mucidospheriums. We were not able to establish a pure culture of this system, but we were able to support it for short periods in a mixed culture containing many other organisms. And at the late stages of development of these mixed cultures, we were able to observe colonies with little or almost no algal cells visible. So these were just clusters of this amoeba crawling across the substratum in pretty the same fashion as previous ones. So our question was, what we are looking at here, and we did extensive search in the literature, the only hint we have found is this source, this book, which shown pretty similar system, and it was referring to small unidentified amoeba infecting Mutidospherium colonies. So what we're dealing with is probably a new undescribed species of amoeba, and it has a strong association with this green algal species. Using single-cell techniques, we were able to obtain some molecular data for these organisms, and using small subunitary DNA phylogeny, we were able to show it belonging to this Granophyllosea clade of Rhizaria, which makes a lot of sense because the other members of this clade are also known to have thin, long pseudopodia with lots of granules. At the same time, this clade is not known to have any symbiotic organisms associated with other creatures, so this would be the first representative having such a mode of life. So this is pretty much what we know about these new organisms by now. So what I wanted to talk about is kind of interpreting these relationships. And this is not as easy as it may seem at first glance, because this finding of the colonies devoid of algal cells suggests kind of parasitoid relationships. So these Mutidospherium colonies can be gradually eaten by amoebas from inside, pretty like parasitoid wasps eating their host, or rather larvae of parasitoid wasps eating their hosts from inside. But at the same time, if you would think about these infected colonies, they act pretty like, I would say, super organisms, the organisms which combine the capabilities of two partners of this symbiosis, the green algal colonies without amoebas cannot move. And of course, the amoebas themselves cannot photosynthesize. So maybe these amoebas are just commensals which hitchhiking these algal colonies, or even beneficial in helping it to distribute in the environment. Either way, this relationship seems quite unusual and remarkable, and of course, it clarifies the phenotype of one of the groups which previously was known only from environmental sequences. So this is pretty much it. I wanted to emphasize that these results were obtained together with Anton Olaf. Also, I want to thank some of my friends and colleagues, and would be happy to answer questions.
Great, thank you. We have plenty of time for questions.
Can you please unshare your slides so that we can switch to another presenter?
Yes.
Thank you.
So I guess I'll start off questions. And so I'm curious about where the algal cells pick up these potential symbionts. Maybe I missed.
Yeah, actually, this is what we would like to know, but we don't. So what we observe in the sample, of course, this thing is not in culture. So we can look at the sample or enriched culture, and what we see is the colonies with the amoeboid cells inside, and a bit later, just these clusters of amoebae. So it is not clear how the colonies are getting infected, but probably these amoebae are capable of some kind of getting inside, maybe crawling inside the colony between green algal cells. I think it would be possible.
Are there any questions in the chat or other folks on? Rebecca.
My question is about getting at the last question you posed. Do you have ideas of how you could measure like fitness of the partners or, some sort of growth components just to see whether like under certain conditions, maybe they're both growing better than if they weren't associating? Or do you have, any thoughts to tease that apart?
Yeah, of course, if we had this system in a pure culture, we would be able to observe it directly, but without being able to do this, I think at least an evidence of the feeding strategy of the amoeba would be really good. And I think the possible step towards it would be doing electron microscopy to see, like, I don't know, is there any phagocytosis happening, or like what is the feeding source for amoebas?